In the last video, we talked about standard addition and why is it important and when is it important to add a certain amount of a standard, which is the same type as the analyte, to your unknown solution, and then start to create a calibration curve based on the addition of the standard. We said that standard addition is important when the matrix interference is important. Now we are going to talk about another type of addition, which is called the internal standard. Internal standard is a known amount of a compound which is different from the analyte. So now we're adding a standard solution of something that is different from your analyte to your unknown solution. The addition of internal standard is important when the signal that we get from our instrument for the analyte is different each time we do the experiment, or is different from run to run. So sometimes for some experiments, for some instruments, you are not getting the same response every time you are using the same concentration of your sample or of your analyte. In this case, you cannot simply create a calibration curve with a different concentration of your standard of your for standard solution of your analyte, and then analyze the unknown and then use the calibration curve to get the corresponding concentration. In this case, we will add an internal standard. The signal from the analyte is always proportional or is always compared with the signal from the standard. The ratio between these two signals compared to their concentration is always fixed, and this is called the response factor. So the response factor is the relative response to analyte and standard. So internal standard will compensate for any changes in the experimental conditions, while the response or while the signal we get from the sample, which is the IX or AX if we are using a chromatogram and we're measuring the area under the beak, varies for the same concentration of your sample, which is X. The internal standard is an added standard which is different from the analyte. To calculate the response factor, we need to have two standard solutions of both the internal standard and of your sample. So in order to calculate the response factor, so you have a certain concentration of your unknown or of your analyte, and this concentration is X. And then you start to run the experiment and you see what is the response to your sample or to your standard solution of the analyte. What is the response? So the response, if it's area under the beak, it's AX. In the same sample solution, we also add the internal standard. So it's only one run. We don't do it in two different runs. And because each will give a different response, each will give a different peak in the chromatogram, then we can simply calculate the response from both the internal standard and from our analyte in the same run. So the response for the internal standard is AIS. This is the response for the internal standard, and IS is the concentration of the internal standard. F is the response factor, and AX over the concentration of X equals F times AIS, which is the response from the internal standard, over IS, which is the concentration of the internal standard. So we first calculate the response factor, and then we use this response factor and rerun the experiment, but now instead of using a standard unknown concentration of your analyte, we use our unknown sample and we add the internal standard in the same manner. And then we see what is the signal that we get from the analyte and what is the signal we get from the internal standard. And because we know what is the concentration of the internal standard and we already know now what is the response factor, we can calculate what is the concentration of our unknown. So let's have an example to see how can we work that out using an internal standard. So in an experiment, a solution containing 0.0837 molar of X here is a standard solution of our analyte. So X here is the analyte, and 0.0666 molar of S, which is the internal standard. So the solution contains both, and this is the concentration of both the analyte and the internal standard. The peak area AX, which is the peak area of the signal coming from the analyte, is 423, and AS is the area under the peak for the internal standard is 347. To analyze the unknown, so this is the first data we get in the first run, and we need to calculate the response factor. Okay, so we're going to use this data to calculate the response factor. Then, to analyze the unknown, we use 10 ml of 0.146 molar of the internal standard. So we have an unknown now. So we don't know what is the concentration of the unknown, but we know what is the concentration of the internal standard. It's 0.146 molar. It was added to 10 ml of the unknown, and the mixture was diluted to 25 ml in a volumetric flask. This mixture gave the chromatogram for which AX, so now the signal we got, or the area under the peak for the our unknown is 553, and the AS is 582. Find the concentration of X in the unknown. So how are we going to answer this question? How can we find the concentration of the unknown? The first thing we need to calculate is the response factor, and the equation for the response factor is AX over X equals F, which is the response factor, times AS, which is the response from the standard, over the concentration of our standard. Now we're going to use the data from the first step of the experiment when we used a certain standard solution of our analyte. So the AX here is 4, 2, 3, and the concentration of our analyte is 0.0837. This is going to be equal to F, which is the response factor, times AS, which is 347, over the concentration of the standard, which is 0.0666 molar. So when you calculate the F here, you're going to find that F equals 0.970. So this is the response factor, and this is the one we are going to use in the next step in order to calculate the concentration of our unknown. So in the second step, we said we took 10 ml of that concentrated of 0.146 molar solution of the internal standard, and we diluted that to 25 ml. So the concentration we are going to use in this equation in the second step is not going to be the concentration in the 10 ml, but it's going to be the concentration, the final concentration of the standard in the 25 ml. So now the concentration of the standard that we're going to use in the second step in our calculation is going to be the concentration of the final standard solution. So it's the initial concentration, which is 0.146 molar, times the dilution factor, which is 10 over 25, because we diluted it to 25 ml. And the final concentration of the standard now is 0.0584 molar. So this is the concentration of our standard. Now, again, let's use this first equation again to calculate the concentration of our unknown. So again, AX over X equals the response factor times AS over S. So it's the signal or the area under the beak for the unknown is 553, and X is the concentration we need to find, will equal the response factor, the one that we calculated here. So it's 0.970 times AS, which is the response from the standard, the final standard concentration, which is 582, divided by the concentration of our standard, which we just calculated, and it's 0.0584. So when you do this calculation, you will find that the concentration of X equals 0.0572 molar. So this is the concentration of the unknown in the diluted solution. So when I ask you what was the concentration of the unknown, I mean what was the concentration of the unknown in the original solution, the 10 ml, not the 25 ml. But this is the concentration in the 25 ml or in the diluted. So this is the final concentration. I need the initial concentration. So again, using the C1 V1 equals C2 V2, you will find that the concentration of the X, which is the initial one, will equal the final one, which is XF, you can call it XF, times 25 over 10. So now when you do, so it's going to be 0.0572 times 2.5, and the final concentration is, you're going to find it's 0.143 molar. So this is the concentration of your unknown in the original solution, in the 10 ml solution. So you have always to be careful which concentration you are using and which concentration we are asking about. I'm never going to ask you about the concentration of the standard and of the sample or of the unknown in the diluted solution, but I'm going to ask you about the concentration in the original solution. So in the same way as you did your calibration curve and your calculation of the unknown in your lab, where when you did create your calibration curve and from the diluted solution of your unknown, you got the concentration, which is the concentration of the unknown in the diluted solution, and you had to work it out to find out what is the concentration in the original solution. So we're going to use the same method here to calculate the concentration of the original solution. For accuracy, we don't just use or run one experiment to calculate the response factor, but we can do like a calibration curve for the internal standard as well. So we use the a standard of our unknown or of our analyte, along with a standard of the internal standard, and we mix them together in different ratios. And then we start to run the experiment several times, measure the response in each run with the mixture. And then we, on the x-axis, it's going to be the ratio between the concentration of the standard, of the analyte, to the concentration of the internal standard. And on the y-axis, it's going to be the corresponding ratio of their responses. Usually, the curve should have the relation should be linear. between these two, and we do that for accuracy. As you see, not all the points are on the linear curve, but we use the method of least square, and then in this case the response factor will be the slope of your, so the slope of the curve is going to be your response factor. So this will help us to guard against any errors, especially the random errors, because as I have been always saying, if there is any systematic error in your experiment or in what you are doing, then you should discard the data point, or you should discard your analysis completely if there is any systematic error. But because there is always a random error, there will be a slight variation from one run to the other for the ratio between the response for the analyte and the internal standard. So this is why we create a calibration curve to make sure that we exclude any random errors.